All right. So we will go ahead and get started. Good morning, good afternoon, and good evening, everybody, depending on where you might be. I am Rob Burgess, and I head up business development for Sino-Biological. And I just want to officially welcome you all to the next installment in our Lock and Key webinar series. During this series, we discuss and hear about cutting-edge technologies which focus really on taking biological research and the development of therapeutics, vaccines, and diagnostic kits, and even basic science to another level. We have a very special guest lecturer today, whom I will introduce to you in just a minute. Before I do that, though, I just want to mention a few housekeeping issues. Number one is we ask everybody to type their questions into the chat box if you don't mind, and we will withhold all questions until the end of the seminar, and then I will walk through those questions as best as I can, try to get to all of them, and I will walk through them verbally with our guest lecturer. And then the second thing is, please tell us where you're from. We love to hear about folks from all over the world and where you're from, so just tell us in the chat box where you're from and say hello. So in that respect, it is my honor and pleasure to introduce Dr. Lauren Nevis, who is with 10x Genomics. And Lauren is going to talk to us today about single cell and spatial multiomics. Lauren is in the Science and Technology Division of 10x Genomics, and she specifically supports biopharma customers in Northern California and Los Angeles. She originally joined 10x in 2023 as a field application scientist, and she brings to 10x Genomics 10 years of extensive molecular biology knowledge across both academia and industry. And just as a further background, Lauren obtained her doctorate from UCLA in working in the lab of Dr. Tracy Johnson, studying co-transcription and specifically splicing and further studies on chromatin structure. And so with that, Lauren, I will turn the podium over to you. And we appreciate you coming today. Just to finish up, the title of Lauren's talk is Better Targets, Safer Therapies, and Higher Success Rates with Single Cell and Spatial Multiomics. Thank you for being with us today, Lauren.
Thank you so much, Rob, and thank you for the fantastic introduction and having me here today to speak. I really appreciate the opportunity to talk about the 10x portfolio and a lot of our capabilities that are really pushing the boundaries of clinical research. And to start off with, I always love this quote from Daniel Davis at the Imperial College in London. The human body has 37 trillion cells. If we can work out what they all do, the results could revolutionize healthcare. And that's really what we're trying to move towards, right? Being able to understand what does every single cell do in the body and how does it interact with other cells and other tissues within the body to really improve human health. And what we've seen is that in the past, traditional drug development tools may be able to offer high resolution or a large scale. Things like the advent of next-gen sequencing has really allowed us to look at a lot of cells, a lot of information at once, but has perhaps not been able to give us the fine-tuned resolution that we need. And this is really where spatial and single cell technologies come in. And so I like to think of single cell and spatial technologies in the context of this analogy that if we think about a single cell as a building, that would mean that organs and tissues are more like cities. A cell is not a static thing. Within a building, there are people, there are parts moving. It is its own individual ecosystem, but an organ or a tissue really involves not just a single building and the parts within it, but how those buildings interact with one another as well. And so single cell sequencing, we can think of as a list of buildings. We're able to, by the shape or by the what is going on within that cell, determine what that cell type is, or in this analogy, what that building is. And that can tell us a lot about a city in this example. We can tell how many commercial buildings there are, how many banks. We can get some idea of how healthy that city is. If there are, 150 Starbucks, but not a single hospital, that could indicate that there's maybe something going on wrong with that city. Um, but spatial transcriptomics offers a better picture of what that city actually is. It can indicate different regions of that city and indicate maybe there's areas that need more service than others. For example, again, if there are regions that have very good access to healthcare, but others may not be, we know which parts of the city to target in terms of helping that area out. And spatial transcriptomics is the same. It gives you that idea of not just is that tissue diseased or not, but what are the portions of that tissue that need to be most addressed. And at 10x Genomics, we have a toolkit, a suite of instruments and assays to help address these issues and help further the learnings that we can get out of a single sample. And so we think of our toolkit spanning from unbiased discovery with our whole transcriptome sequencing-based technologies with the Chromium single-cell assays and our Visium whole transcriptome assays, all the way to our more focused spatial insights with our Xenium platform. Xenium is a fluorescent-based output that uses smaller, very, very high resolution, actually subcellular resolution panels so that you can get the highest resolution and discrimination between transcripts. And just a little bit more information about those individual assays, or I'm sorry, about those individual platforms. Again, with Chromium, we have a lot of different modalities within that single cell platform, not just gene expression, but also looking at things like B cell or T cell receptor sequences for antibody discovery, CRISPR screens, ability to look at both protein and RNA expression. Probably my favorite, which is a chromatin accessibility based on my background. And again, this is a sequencing-based technology. Similar to that, we have our Visium assay, which I mentioned is also a sequencing-based technology, but this actually pairs that transcriptomic information directly with histology and with tissue information overlaid on top of it. And then finally, Xenium, as I mentioned, is a panel-based assay. This is a fluorescent output, so there's no sequencing required. And we are able to look at anywhere from a few hundred up to over 5,000 targets at the same time, directly on the tissue itself. And these tools are really quickly becoming critical for clinical research, everywhere from early discovery to post-market surveillance. So we are able to use this to identify and validate targets within cells, mapping cell types and pathways involved in disease. During clinical research, we've seen that we're able to identify therapeutic MOA, mechanism of action, as well as be able to evaluate toxicity and biodistribution. And so I'm going to take a little bit of time to talk about each of those platforms in more detail and really where they can help with some of those steps in the clinical research or the drug development process. Just as a quick level set, single cell sequencing has been around for a while, but I like to start with a high level overview of what does that data actually look like. And here we have an example of what a single cell transcriptomic output from a renal cell carcinoma sample looks like. So in this case, we're looking at a UMAP, which is a projection of so much information on a 2D scale. So each of these individual dots represents a single cell, and they're clustered and color-coded based on similarities in gene expression profile. And those gene expression profiles can help us actually determine what those cell types are and be able to annotate those cell types and determine relative populations. But it doesn't stop there. We can definitely go deeper, looking not just at those high-level cell types, but also more fine-grained cell types and gene expression. So in this case, we were able to look at those T cells in more detail and be able to distinguish and determine differences between the CD8 effector T cells, the regulatory T cells. And you can imagine that if we're comparing this directly to a healthy population, we would then be able to look at the differences in gene expression of particular marker cells, marker genes between those two samples. And so how can clinical research, how can translational research benefit from these single cell analyses? There's a lot of ways. One that I already kind of mentioned, cell heterogeneity, being able to identify rare or unknown cell types, those mechanism of action studies, looking at the pathways that are affected in disease. I'll talk a little bit more about antibody assessment actually in a few slides, biomarker discovery, and then of course looking at toxicity or immunogenicity markers. And in fact, this medRxiv, this preprint came out late last year that indicated that drug development really, really highly benefits from single cell research. And in fact, they found that targets that were supported by single cell RNA sequencing were 39% more likely to move from phase 1 to phase 2 trials. And one example that I want to show, this is a little bit of an older example. This is from 2018, but this was really important for actually how we think about addressing cystic fibrosis and how single cell sequencing really made that shift in focus. So up until 2018, a lot of prior research on cystic fibrosis focused highly on ciliated cells. And what this paper actually found was that there was actually this very small population that had been previously unidentified with bulk sequencing that they termed ionocytes. And these were important because they actually contributed to more than 50% of all of the CFTR expression within the lung tissue, that gene that is often mutated and often very, very key in driving cystic fibrosis progression. And it turns out that this really wasn't very present or very well expressed in ciliated cells. It was really in those ionocytes. And so we were able to now shift how we think about addressing and targeting markers and individual targets within those ionocyte populations to better improve cystic fibrosis therapies. So bringing us around to the families of chromium single cell assays, we do at this point have quite a few assays available that span a lot of different applications, but they broadly fall into three major families. Our universal assays, which are going to be reverse transcription based, our flex assays, which are probe based, and then our epi assays, which that is short for epigenome, which combines gene expression with chromatin accessibility. Regardless of which assay you choose, they're all going to use very similar streamlined workflows where you're starting with a single cell or single nuclei suspension that gets pipetted into our microfluidic chips that allows for automatic partitioning of those cells. That is then followed up by a couple of relatively simple steps similar to bulk RNA sequencing to allow us to actually be able to sequence that data. And then we also have a suite of analysis tools called Cell Ranger and Loop Browser that are available for free for you to pre-process and analyze that data. A couple things that I'll touch on a little bit more later in the talk too. We are now able to analyze up to 20,000 cells per sample, although you can actually push that quite a bit more with multiplexing. And we're able to multiplex up to 128 samples per chip now. And you'll see actually we're about to release a new product in just a week or so that is going to allow you to go much farther than that. I currently can't do the math right now, but it's 384 per channel. So whatever 384 samples times 8 is now what we're going to be able to run on the chip. So massively pushing that scale. So digging in a little bit more into those product families here, I'm comparing and contrasting our universal assays versus our probe assays. So once again, those universal assays are reverse transcription based. They are species agnostic within the eukaryotic space because they do rely on polyadenylated reverse transcription of polyadenylated transcripts. This also means that you're actually capturing full-length cDNA. So this is compatible with long-read sequencing, isoform analysis, SNP analysis, et cetera. So very, very flexible in terms of what kinds of sequences you can look at. Our flex assays, on the other hand, are probe-based. So instead of pulling down the RNA directly, we are hybridizing probes to the RNA and then pulling down those probes. And this makes it highly flexible in terms of being what sample types you can work with. You'll see that these are compatible with a broad range of input samples, including fixed cells, FFPE, fixed whole blood. And we've also seen that because it is probe-based, it is less reliant on full-length intact RNA, which means that it does perform better with low-quality samples. You'll also see in a moment that this is going to be our lowest cost at scale assay. And this is also the one that I mentioned you're going to be able to pool up to 384 samples at a time. So multiplexing options are available for both of these universal and flex assays. So I already touched on the flex assay a little bit, but just a little bit more detail about that. Because we are pulling down probes instead of the RNA directly, we have the opportunity to actually incorporate a sample-specific barcode into those probes. And that's really what allows us to multiplex at high levels and be able to demultiplex down to the transcript level based on that transcript probe. We do have another option as well for the universal assays. In this case, the method with which we're multiplexing is a little bit different because we're actually multiplexing on the chip itself, making use of some specialized microfluidics to multiplex up to 4 samples at a time without any kind of pre-processing, any kind of upfront sample handling. So, oh, my animations didn't work. That's okay. These assays cover a wide breadth of sample types as well as multiomics. So you can see on the left-hand side, we have our list of several different examples of just the sample types that we have worked with. But I want to draw your attention to the right-hand side where we discuss the number of multiomics that we can pair with gene expression. And so I'm going to talk about a handful of these in a little bit more detail, starting with that T cell and B cell receptor sequence analysis. So I showed you this before, where we're able to look into cell type analysis of these T cells. With our 5' gene expression immune profiling assay, we're actually able to look at the alpha and beta chains or heavy and light chains of T cells and B cells respectively, and actually be able to look at those individual clonotypes and their concentrations within a sample. And so in this nice example publication from 2020, these authors profiled both gene expression and TCR sequences of more than 100,000 T cells from various cancer patients, including lung, renal, endometrial, and colorectal cancer. And they were able to find that that CD8, dual CD8 expansion in both the tumor cells and the normal adjacent tissue predicts better response to immune therapies. And so this is really allowing us to now use peripheral blood as a less invasive biomarker and patient stratification study for these particular cancers. Additionally, here we're actually looking at B cell receptor sequences for antibody discovery. This one was looking at more than 25,000 B cells across hybridoma lineages, and they were able to identify um, 700 over 700 new antibodies and new lineages, indicating that with this technology, we can really accelerate candidate optimization by being able to rapidly identify those new antibodies. I'll also mention for these assays, we have really started to focus on automation. I know that automation is going to be really key for fast turnaround and being able to drive a lot of those studies forward. And so we have validated methods on both the Beckman Biomech i7 as well as the Hamilton NGS Star MOA to automate library preparation. Another key point that I want to address here is our compatibility with FFPE tissue. This is driven by our GEMX Flex assay, that probe-based assay that I mentioned. This assay is not only compatible with fixed cells and fixed nuclei, but this also allows you to fix tissue directly. So in this case, you could actually take a small tissue piece, chop it up, fix it with 4% paraformaldehyde, dissociate it at a later time when you're ready to run it. But in the meantime, it allows for easy storage for up to a year at -80 with no changes to the gene expression profiles. And then what I want to focus on the most in the coming slides is actually our compatibility with FFPE. FFPE, we know, can be difficult to assess via RNA sequencing because the RNA does tend to be a little bit degraded during the dissociation process, and those poly A tails are not always available. This assay with the probe-based analysis allows us to assess those types of blocks in high resolution. And then finally, I'll just mention, we are also able to analyze fixed whole blood. So not isolated PBMCs, but actually whole blood fixed with paraformaldehyde, which really opens up the ability to work with clinical sites. And it does not require that expertise to be able to isolate PBMCs on site. They can fix the blood and ship it for isolation later on. So this paper that came out last year, really, really nice paper that was actually comparing our Flex assay with SNAP frozen tissue versus FFPE. And what they found was it generated very comparable data, really highly consistent between the split samples that were embedded in FFPE versus the ones that were SNAP frozen for our Flex assay. And again, this really helps with that long-term clinical follow-up data. And this also means that FFPE blocks that might have been generated a while ago, or you may not have access to fixed or frozen tissue, are now able to be analyzed using single-cell data. I do want to touch a little bit more on the multiplexing capabilities of Flex, because this is really driving down the cost of single-cell sequencing, again, because of that sample-specific barcode that enables inline multiplexing. So this is an example where we actually ran, I believe, a full 16 samples. I'm not showing the data for every single one. But in a single channel, a single library, and we're able to demultiplex that. And this costs less than a cent per cell to be able to run that. We aren't stopping there, though. As I mentioned, we are expanding our multiplexing capabilities. We are now open for pre-orders for our GEMX Flex V2 assay. And we are planning to start shipping that within the next couple of weeks. And this is going to enable massive scale, so up to 1 million cells per channel or 8 million cells per run. That means being able to automatically partition 8 million cells in less than 6 minutes. And then not only are you going to be able to pool up to 384 samples, you actually have the flexibility to pool as many cells, or I'm sorry, as many samples as you want. So we don't limit you to being to needing to run that full 384 samples. You can run 2, you can run 96, you can run 150. You pick and choose, and we see this as going to be very, very useful for groups that may not be running the same number of samples every single time. And then finally, I love pointing out this publication, or this preprint rather, from the Fred Hutch Institute that actually looked at 10x technologies, single cell technologies, compared to a number of other technologies on the market, and found that our assays, the flex assay, our universal assays, won out on almost every single metric, sensitivity, scale, being able to identify cell types. And they really were impressed with the robustness, the sensitivity, and cell recovery of our assays. So I have a link here if you'd like to take a look at all of the different metrics that they analyzed. Switching gears a little bit now to spatial technologies, we really do see this as working cohesively with single cell technologies, but again, being able to give us that map of the tissue, being able to identify those cell-cell interactions with more detail, and be able to determine not just the health of the tissue, but also looking at the microenvironments of the tissue. So again, we can see that not only we're able to look at precise cell types, but cell type characterization, target microenvironment characterization. This is also really helpful for looking at toxicity or immunogenicity of not just the immediate target cells, but also surrounding cells, and how widespread are those responses. And just to reiterate, we have 2 complementary technologies for spatial analysis: our Visium unbiased assay, which is looking at whole transcriptome gene expression via sequencing, and then our Xenium for precision insights down to subcellular resolution using a fluorescent-based output. So starting off with Visium, this was the original spatial technology from us. If you are familiar with Visium but have not taken a look in a while, I do encourage you to take a look at some of the new data sets that are coming out, because we are now able to offer gapless coverage of a tissue at single cell scale. If you recall Visium in the past and some of these other spatial technologies, we were able to look at maybe several spots of a tissue, or there might have been gaps within the tissue, but now Visium HD allows for complete coverage of our target tissue. And I'll point out too, this is not color-coded or using IHC. This is actually just the default unsupervised clustering of these this colorectal cancer sample directly output from the Visium analysis. How is this being used? This is a fantastic publication looking at idiopathic pulmonary fibrosis. And one of the things that I really like to point out with Visium is, because you're looking at intact tissue and looking at tissue slices directly, it allows you to actually recover a lot of fragile cell types that might have otherwise been lost during dissociation. In particular, they found that there was a much higher concentration of epithelial and endothelial cells that they suspected they might have been losing during single cell dissociation. Not only that, but they were able to identify disease-specific niches within this fibrotic tissue that really helped determine the cell-cell interactions between these cell types. Just a quick overview of that workflow. Again, we're starting with a tissue slice directly. So we're actually overlaying that gene expression information directly onto that tissue. You'll see in a moment what the capture slide looks like, as well as the instrumentation that we're using to improve capture of those analytes. And then after capturing those specific analytes, the downstream workflow is, again, very similar to any other sequencing library, and very similar to a lot of our single cell sequencing libraries as well. So Just a little bit more information on that capture slide, this Visium capture slide. This is a 6.5 by 6.5 millimeter capture area. There are 2 per slide, and it is covered with a lawn of 2 by 2 micron squares that include this capture oligo that allows us to capture either probes or polyadenylated RNA. And includes this spatial barcode that tells us exactly what 2 by 2 micron bin is it coming from. There is also flexibility with the resolution that you can look at your tissues as well. As I said, the squares are 2 by 2 micron bins. That tends to be smaller than most cells. So we automatically bin by 8 by 8 microns. But you do have the flexibility to choose and optimize whichever bin size is most appropriate for your tissue type. In this case, we are not sectioning directly onto that Visium slide. With the Visium CytAssist instrument, we are actually able to use tissue section directly onto glass slides, which makes the sectioning, for one, much easier because you don't have to try to navigate it into an individual capture area. You can place it almost anywhere onto a glass slide. And when it is sandwiched with that Visium slide, it will be placed onto the capture area. This also means that you can use archival slides. If you don't have a FFPE or OCT block available, but you do have tissue sections that are already placed on glass slides, you are able to use this with Visium using the CytAssist instrumentation. Not only that, but this technology it really helps control capture of those analytes. So it really does prevent any targets that can be released or dispersed. That was sometimes the case with direct placement onto slides. So I mentioned analytes, and that's really because similar to our single cell sequencing assays, Visium HD now offers 2 different chemistries. Our probe-based assay, which is very, very similar to the Flex assay and actually uses the same targets for mouse and human. And our Visium HD 3', which again uses our 3' polyadenylated assay technology. This one is relatively new. It came out just a couple months ago, and we have already tested on a lot of different tissue types, which you'll see in a moment. So again, probe-based assay for human and mouse. The targets are going to be the same as our Flex assay. So you can actually pair the Visium single cell, the Chromium single cell and the Visium HD assays for true single cell integration. Our 3' assay now, of course, because it is species agnostic and pulling down any polyadenylated mRNA, we expect to be used in quite a number of ways. In this case, one of the key asks that we've had from a lot of our users is the ability to look at xenograft tissues. And now you can do that with high fidelity, being able to identify the host versus the xenograft tissue within a sample. I love this slide just because it shows all of the work that we've done internally across different tissues, including things like pig, canine, lots of non-human primate samples, chicken. I'm not showing it here, but we have also validated this on plant tissues. I'm not sure if that is of interest of anybody on the call. It tends not to be as much of a clinical focus, but we have also looked at plant tissue now. Finally, but certainly not least of all, is our Xenium assay, our Xenium in situ analysis, which is the newest instrument and newest assay that we have, which enables very, very precise subcellular resolution of transcripts. So here we're looking at a tissue that is, we have identified the individual single cells using a cell segmentation stain that is incorporated into the Xenium workflow, that when it's paired with our machine learning algorithm on the instrument, is able to identify individual cells with very, very high accuracy. And so you can see that it's even able to identify these smaller cells versus larger cells in the periphery. And then when we zoom in, you can actually see the output from individual transcripts and get localization, not just down to what cell is it in, but also is it in the cytoplasm versus the nucleus of these cells. So this is a publication that we actually put out last year. I apologize, this slide is a little bit out of date. It is now in Nature Communications and no longer just a preprint. But in this case, we used one of our Xenium panels, the human colon panel, with an additional 100 custom genes that allowed us to not just look at typical gene expression, but also distinguish between T cell clonotypes. In this particular sample, we were able to, using those clonotypes, identify an expansion of anti-tumor T cells around these foci of particular chemokines in the tumor microenvironment of these colon cancer patients. Not only that, but we've seen that Xenium is now being used quite a bit to look at spatial biomarkers, particularly for cancer immunotherapies. In this case, these authors used the human breast cancer panel or human breast panel in order to look at cell organization and cell-cell interaction that are involved in PD-1 blockade response in these breast tumors. And what they found was the spatial proximity of the cytotoxic T cells versus the actual tumor cells was critically important for predicting how well a cancer sample is going to respond to immunotherapy, which realistically makes a lot of sense. You do need those T cells to be close to their targets in order to be able to actually carry out their function or to be able to overcome T cell exhaustion with the help of some of these immunotherapies. But until these spatial technologies, we weren't able to actually look at that and determine those spatial interactions at that high resolution. So Xenium does have a simple and fast workflow. In my opinion, having run all of these personally, this is actually the easiest one to run in terms of upfront tissue handling and staining. You start off with a Xenium slide and section tissues directly onto that slide. It's followed by probe hybridization. And I do want to point out here, these probes are what really drives the sensitivity and specificity of the Xenium assay. You can see that these are actually padlock probes in which each side of that probe is required to fully bind its target in order for it to be ligated together and output any kind of signal. So that's what really allows us to look at down to single nucleotide differences. We're able to look at SNPs, isoforms, small mutations, clonotypes because of that padlock probe chemistry. Additionally, we employ rolling circle amplification that allows us to get down to that single transcript sensitivity. That is then followed by that cell segmentation stain that I mentioned, and then it goes on to the Xenium analysis instrument. And this can take 3 to 6 days, but it's totally hands-off time, and that 3 to 6 days is variable depending on how many targets are you looking at and how big is the tissue. But what I like to point out is that because it's a fluorescent technology, the data is available the moment the run is done. There's no need to do any other pre-processing before you can actually look at that data. And that's because the Xenium analyzer performs not just the imaging and decoding of individual transcripts, but it also automatically segments those cells, assigns cells based on those transcripts, and even does unbiased and k-means clustering before the run is complete. So when the run is done, you're able to actually take a look at this directly on the screen of the Xenium instrument or on your desktop using our free Xenium Explorer technology. And here there's just a quick example video of what that data looks like. So we're zooming in on these samples here and able to look at how many transcripts are included for each transcript type and for each cell type. I also like to point out that Xenium is actually non-destructive. The chemistry that we use means that the tissue is still intact after the run is done, enabling you to perform other analyses on the exact same tissue slice. And so we have this nice demonstrated protocol and technical note that describes how you can carry out immunofluorescent staining, H&E staining, or you can even pair it with our whole transcriptome Bayesian analysis. So perhaps you're looking at a large tissue slice and you want to identify some regions of interest based on some high-level markers, but then you want to gain more insight about the entire transcriptomic profile. You can do that by following up on that section of interest with the Visium analysis. I've mentioned a couple already, but just a little bit more detail about those panels. We do offer a number of pre-designed panels that are within the several hundred gene range for human and mouse. These have been pre-designed and validated in-house internally, and you can see the list of those here. All of these, you can actually add on up to 100 custom genes. So if you take a look at the panel and say, this is great, I like most of these, but I have a couple other targets of interest to add to that, we can definitely do that. We also offer a 5,000 plex panel for human and mouse that has been chosen based on actually a lot of various publications that indicate what genes are best for cell markers, pathway determination, disease descriptions, and things like that. Similar to those smaller panels, you can also choose to add on 100 extra custom genes onto that panel. We also offer standalone custom. So if you want to create a fully custom panel or you're working with non-human or non-mouse species, we can definitely do that. We've had a wide variety of organisms that we've worked with already. And then I've touched on this, but just to reiterate, we also are the only ones that are able to offer advanced custom design against things like single nucleotide variations, isoforms, and exogenous sequences. So I've started to see a number of people, especially those who might be looking at LNP therapies or any other kind of gene therapy deliveries. You can actually look at exogenous sequences and cargoes that have been carried to your tissue of interest by pairing the advanced custom target with one of our panels. Also, just released, we now are not only able to look at RNA gene expression, but also protein gene expression with our new immuno-oncology protein panels. So these are 27 total protein markers that come in 7 modules. So you can run these individually or run all of them at once, depending on what best fits your project needs. We've optimized this across a number of different tissues. It adds on about an hour's worth of incubation time in the upfront workflow. So very, very minimal workflow changes and allows you another layer of information on that exact same tissue slice. So in this case, we're again looking at a cancer sample. In this case, I believe it's a clear cell renal carcinoma. That we had annotated by a pathologist, but then also compared it to both RNA-based clustering and protein-based clustering. And what I like to point out here is that there is this region of cancerous cells that via RNA looked very, very similar in terms of their transcriptomic profiles. But once we looked at the protein-based clustering, we saw that there was a region of these cells that actually had a different protein expression pattern, indicating that there might be some post-transcriptional regulation occurring in some of these cancer cells, indicating some changes or differences in progression of those cancerous regions. And in the last few moments, I just want to follow up with one more example because it's just this fantastic example of how xenium is already being used in clinical trial studies. In this case, this was the Deuterio trial. This one was actually carried out as a retrospective, and that's because this was looking at a bladder cancer immunotherapy that originally they stratified their patients using a bulk RNA sequencing analysis called the tumor inflammatory signature, which has been validated for other cancer types, but not for bladder cancer. Again, it's a bulk RNA measurement, so it's just looking at activated but suppressed immune responses in the tumor microenvironment, but does not create the spatial resolution that other spatial technologies can provide. And unfortunately, what happened was that within this clinical trial, that tumor inflammatory signature did not accurately predict response to immunotherapies, this Deuterio therapy. So they went on to test a number of different assays, including single cell RNA sequencing, several other spatial technologies. And what they found was xenium actually best predicted or found spatial biomarkers that best predicted the response to this immunotherapy. And they did this by not only identifying cell types, but also building descriptions of cell communities. So with spatial technology, again, not only identifying what those cells are, but able to look at the interaction between those cells, what cells are closest to one another, in what is often called community or niche analysis. And what they really found was the relative abundance of cell types in these communities in responders and non-responders indicated which ones were going to respond best. And so in communities or in patients that responded, they showed these communities of cell types that included dendritic cells, M1 macrophages, CD8 T cells, T regulatory cells, and NK cells. Essentially, there were effective immune populations that were embedded in and close to those cancer cells so that they could actually carry out their function against those cancer cells when supplemented with the immunotherapy. So really great example of how this is already being used for things like patient stratification. And so that does bring me to the end of my talk. Before we stop for questions, I just want to hand it back off to Rob to describe some of the cynobiological portfolio.
Thank you, Lauren. That was a wonderful presentation. We really appreciate you taking the time to put it together and presenting it to us. Yes, I also appreciate this final plug for cynobiological. I'll just very briefly go over these two slides. Starting in the upper left-hand corner, Cyno does have almost 9,000 what we call full-length recombinant bioactive proteins made in mammalian cells. We actually have the world's largest collection of mammalian cell-made full-length bioactive proteins made primarily in HEK or 293 cells. We have also chosen the best-selling targets for those proteins, as well as some strategically chosen drug targets and diagnostic targets, and made almost 15,000 either monoclonal, polyclonal, or recombinant antibodies, which bind to those. And related to that, we have about 700 ELISA kits finished related to those targets. We also have, through our acquisition of Signal Chem Biotech in May of 2024, about 1,500 active enzymes. These include kinases, phosphatases, histone acetyltransferases, HDACs, and other epigenetic-related enzymes. And so that is, in fact, the world's largest collection of active enzymes and certainly kinases for offer to the research community. In addition to that, we have a line called Provir, which is our line focused on viral antigens primarily, as well as corresponding antibodies that bind to those antigens. So not certainly COVID-19, but also SARS-CoV-1, HCV, Hep C, RSV, HIV, and many, many other viruses. It's a huge collection of antigens and related antibodies. So please look into that if you're a virologist or an immunologist. And in the lower part of this slide, we have custom services related to all of these types of products. So if you have a transmembrane domain protein or a difficult enzyme to manufacture, please allow us to design, develop, and manufacture that in a custom format for you, as well as the corresponding monoclonals, polyclonals, or recombinant antibodies. And in fact, we can even do enzyme validation and develop custom assays for you as well. And then over on the right here, you can see that we do have endotoxin-free proteins made right here in Houston, Texas. And I have that on the next slide, but also I also mentioned the active kinases and enzymes, which are made in Vancouver and are part of our acquisition of the wonderful company Signal Chem Biotech. So if we could go to the final slide. I'll just emphasize a little bit more about our endotoxin-free production in Houston. See, we have 8 minutes left here. So when we say endotoxin-free, what we mean is below the lowest level of accurate or reasonable detection of endotoxins. So this includes vectors and plasmids for transfection into cell lines, for the production of endotoxin-free proteins and recombinant antibodies, the buffers as well, along with they've been ultra filtered, and those are available endotoxin-free and very low endotoxin level plasticware. And in fact, our instrumentation in Houston has regular clean in place capabilities and methodologies. So we do not have to disassemble and manually clean our instrumentation when putting together these reagents or the final endotoxin-free protein product line. This is known as our ProPure product line. It is made exclusively in Houston, and it includes off-the-shelf products as well as custom made-to-order proteins and recombinant antibodies. You can look on the Sino website and you'll see the ProPure collection made here in Houston. For the custom ones, we can actually develop, manufacture, and deliver custom endotoxin-free proteins and recombinant antibodies in about 3 weeks or less. And then we have up to 50-liter cultures per batch, which should produce hundreds of milligrams. And we do HPLC verification on all of our ProPure product line. And so with that, again, Lauren, thank you for allowing me to plug Sino for just a few minutes at the end of your talk. So,
Thank you very much. Were
yeah,
there any
yeah.
questions?
I was just going to say at this point, we'll turn to the questions in the chat box. We have a few minutes left here. And so I ask anybody attending to type in a good question. We do have one question we'll start out with. This is from Bahram Badlu. I believe he's based in the Netherlands. And it's a very broad perspective type question regarding the technology and its evolvement over time. Bahram mentions that the core technology that 10x is based upon is about 20 years old, and I believe it's based upon some technology developed in 2005 called fax tissue, if that's correct. And he just wanted you to comment on how it's evolved over the last 20 years and really what sets 10x apart from the base core technology and your competitors, I guess.
Got it. Is this for, I apologize, I'm not actually familiar with fax tissue. I assume that's also a single cell technology based on the fax portion of that, but that's not actually one that I'm super familiar with. I'd say the key things that 10x has really been driving is the scale at which we're able to do things within a usable workflow. So some of the early single cell technologies, you may have only been able to look at a relatively small number of cells, kind of started off by really just dropping individual cells into a 96 or 384 well sample. So you can, or well plate. So you can imagine that if you were trying to look at 8 million cells at a time, which we're now able to do in about 6 minutes, that would take a very, very long time to do with 384 well plates. And so being able to automate that partitioning and be able to do that at scale has really helped us identify a lot of those very rare cell populations that we might not have seen before. And especially now we're seeing the advent of AI being very, very important for a lot of different aspects of clinical research. And that many of those models are being built on single cell technology, and a lot of them are being built on 10x single cell technology because you need a lot of information across many, many different sample types and many, many different cell types to be able to feed into that MLM and be able to create those models. So scale is a huge one. And then the other one that I always like to point out is just the additional modalities that we've added over time in a way that's been validated and very, very heavily tested. So again, being able to pair things like gene expression with, I didn't get a chance to talk about it, but chromatin accessibility, protein analysis, all paired within the same cell as opposed to having to bioinformatically combine those together.
Mm-hmm. Great. Thank you for that answer. I appreciate it. I realized it was scale, automation, capacity, things like that. Of course, you've probably taken the sensitivity to another level as well. So kudos to y'all at 10X. Just a real quick question I had, is your system a closed system where like all the slides and the reagents, consumables, and buffers, I guess, have to be purchased from 10X along with the instruments, or is it more open? Do you have partners that sell the consumables?
So it does depend a bit on what instrument it is. Something like the Xenium, because that is kind of an all-encompassing instrument. Um, Most of those consumables are going to be produced and will need to be purchased from 10x because they do fit very specifically onto the deck that we've made. Something like Visium, where we do have those specific Visium slides, those kinds of things that, again, work very specifically with that instrument need to be purchased from 10x, but you are able to incorporate your own glass slides. And there are a number of workflow steps upstream that if you wanted to add on other modalities, those could be purchased from our partners. And then single cell, I would say, again, the microfluidics going on to the Chromium instrument that are doing the partitioning, that is a 10x technology, so that's from us. But many of these multiomics that you're looking at, like the intracellular protein, like the cell surface proteins, the CRISPR guides, those are all ones that we do partner with other vendors and other users, and it's fairly open in terms of what can be used as long as it is compatible with our downstream assay.
Great. Thank you for that answer. I think we'll go ahead and wrap it up on that. And Bahram, I want to thank you for that great question. I do want to say we had a great turnout today. We had folks from all over the United States, as well as from the Netherlands, Singapore, and the UK. So I want to thank our attendees for attending today. Lauren, I certainly want to thank you. It was a fantastic presentation, and kudos and congrats to you and everybody at 10X for all your success that's already occurred and will occur in the future. And then finally, my colleague and one of our newest employees at Sinobiological, Ms. Kayla Sarmiento, who helped execute this webinar and seminar today. We had some hiccups at the beginning, and Kayla came through in flying colors and made sure that we got started on time. So thank you, Kayla, for that. And in that respect, I guess we'll sign off, and I wish everybody a good afternoon or good evening. So thank you again.